Good morning, ladies and gentlemen. Welcome to the AVACTA Group PLC full year results presentation for the year ending 31st of December 2020. Throughout this presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted any time via the Q&A tab situated on the right-hand corner of your screen. Simply type in your question and press send. With over 1,500 investors having accepted to attend today's presentation, the company will not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it's appropriate to do so. These will be available via your Investor Meet Company dashboard and you'll be notified once they're ready for your review. I'd also like to remind you that this presentation is being recorded. Before we begin, we'd like to submit the following poll. I'd now like to hand you over to Elisa Smith, CEO, Tony Gardner, CFO, and Neil Bell, Chief Development Officer of AVACTA. Good morning.
Thank you very much, and good morning, everybody, and thank you for sparing some time for the results presentation. We are quite limited on time, so I will get cracking straight away. There's a lot to get through. So we've already been introduced. Myself, Tony Gardner, and Neil Bell will be presenting to you today. I think many of you will be familiar with the company, but just let me give a very brief background. AVACTA Group has two principal divisions: the diagnostic division based in Yorkshire near Wetherby, around 50 individuals, scientists and support staff focused on diagnostics development, with a large number of partnership collaborations, technology evaluations ongoing around the world. Of course, we will focus primarily today on the SARS-CoV-2 rapid antigen test, which is at the front of everybody's minds, I know. The therapeutics division based in Cambridge, UK, around 35 staff there. Neil has just established a clinical development group, which I'm sure he'll talk about in a later part of the presentation. Focus on oncology, cancer therapies, using our two proprietary platforms, the AFMA platform and the Precision platform. And again, a number of global partnerships in oncology, autoimmune, cell and gene therapy, which we have limited time today to go through all of those, but we will focus on the AVA6000, the precision prodrug form of doxorubicin, which we will take into the clinic this year. So let me start with the preliminary results for period ended 31st December 2020. And Tony, would you mind taking us through the next few slides, please?
Yeah, sure. Thanks, Alistair. As from the RNS this morning, the main operational highlights around the therapeutics business are the AVA6000 Pro doxorubicin clinical trial and the planned dosing of patients in phase 1, which Neil will cover later on in this session. Alistair will also touch on the partner programs with Daewon and LG Chem, and also the newly announced deal with Point Biopharma. In terms of the diagnostics business, obviously the main focus is around the antigen lateral flow test, which we'll go through in detail after the financial highlights, but we'll also pick up in terms of the recent deal with Australia Bioseparations that Alistair will cover later on. In respect to the financial highlights, we'll cover these in a bit, but just to pick up on the fact that Paul Fry was appointed to our board as a non-exec at this time last year. He's the CFO of AVACTA Group, and he is a good addition to the board and chairs our audit committee. In terms of the financials that were reported, just to make it clear, the results are for the 12 months ended December 2020. The comparative period on the slides there is for the 17-month period ended December 2019, as we changed the year end to a calendar year end. The revenues are reported at 3.6 million, which is slightly down on the previous year. But as you would appreciate, within both the diagnostics and therapeutics business, anything kind of non-COVID related workflow has been slightly restricted given our ability to get all the scientists back into the labs, but also the 2019 number included an upfront technology access fee from LG Chem during that period. The R&D costs are the main focus of our business, and the 10 million reported there covers the all the costs of the therapeutic development programs progressing AVA 6000 through towards clinical trials, but also the development costs around the lateral flow tests that have been incurred during 2020. The retained losses for the year are just shy of 19 million compared to 15.6 million the prior year. In terms of the balance sheet and cash flow, we obviously completed the fundraising during last year, which raised a gross 53.8 million. That will fund the current business plans, excluding any COVID revenues from the lateral flow test, well into 2023, and is consistent with our announcements with the fundraise in terms of where we envisage the cash flow and how we manage that across the programs. The cash at 31st of December was just a fraction under 48 million compared to just under 9 million at the prior year end. So we are well funded to deliver what lays ahead in 2021 and beyond. I'll hand over to Alistair, who will bring you an update on the diagnostics division.
That's great. Thank you, Tony. Okay, so before we get into the detail of the rapid antigen test, I've been through this with some of you, I'm sure, before, but I wanted to just give the background again to the use of lateral flow tests and PCR tests. So what you see in front of you in this diagram is the evolution of the viral load. So assuming an individual's exposed to the virus, the red line shows the increase in the amount of virus in the individual over time, over several weeks. The period during which the individual is infectious and then shows symptoms at the peak of that curve. And then superimposed on that are two lines, a solid line that is the lateral flow test detection limit, and a dotted line below that, a more sensitive PCR detection limit. And really this is key to the correct use and the complementary use actually of lateral flow tests and PCR, which I think has been poorly communicated. Certainly last year, it's getting better now, but poorly communicated, which has led to a perception that lateral flow tests don't work. So the correct use of a lateral flow test is for frequently screening individuals, large numbers of individuals, which is not possible from an economic or even a capacity perspective with PCR. And what you see on the lateral flow detection limit line is frequent testing of this individual with negative results until the point where that person has a viral load that is infectious. And of course, that is the point at which you want to identify the individual to be able to isolate them and reduce the risk of spread of the disease. I'll talk a little bit on the next slide about what infectious really means. You can see that there is a little window there where PCR could pick up that infection sooner than that point, because PCR is more sensitive. But the downside as well of PCR, if you look in the post-symptomatic, post-infectious time, PCR will still pick up that lower viral load, which is not an infectious viral load. It will still pick that up and give a positive result. And if you were to isolate that individual, then there's an economic cost which doesn't need to be borne. So these are complementary testing methodologies that combined give you tools to reduce the spread of an infectious disease like the SARS-CoV-2 coronavirus. Okay, so this is biology. Okay, so there isn't a black and white definition of what infectious means. The viral load that the individual carries. You then have to start to think about the amount of exposure that somebody gets to them, the type of exposure, whether it's in a plane for 8 hours or on a bus for 5 minutes, or more intimate, if I can put it like that. So there are clearly different scenarios, and there isn't a black and white level of viral load that makes you infectious. But we need to just try and put some figures around this. And the benchmark, of course, is PCR. And PCR results are given as what are called cycle time or CT values. And just as a reminder, the lower the cycle time value, the higher the viral load, so fewer amplification cycles required to detect the virus, and the lower the viral load, the higher the CT value. So what you see on the left of this figure, the histogram, is the likelihood of infecting one of your contacts. So this is track and trace data from a study in Liverpool just before Christmas. And that likelihood of infecting your contact is color coded depending on the amount of virus that the individual carries. So red is a very high viral load, shown there as over 100 million copies of the viral RNA per milliliter of sample. Amber less, blue less, and so forth. And you can see the corresponding PCR-CT values on the right-hand side, 2 columns, one measurement made in Porton Down, one measurement made in Glasgow University. So you can also see the variability there in CT results. So you should be constantly thinking biology is not discrete numbers, it's a little bit more variable than that. But we can put a broad line in there. This particular publication that I'm referencing suggested a CT of 25 as the 25 and below as the infectious level by PCR. I would suggest a slightly higher CT value than that. A lower viral load could still be infectious if you have prolonged exposure, or depending on the nature of the exposure. But you can see that once you get down to that level, the dark blue bar, the likelihood of infecting a contact can be as low as 10%. So being able to pick up CT values of 27 or below is a good is a good indication of infectious. So let's bear that in mind as we go through the performance of the test. So what I've done here is I have put on this slide the formal regulatory intended use of the test. So it is a qualitative test for the SARS-CoV-2 antigen using anterior nasal swabs. So if you weren't aware, our AffiDX antigen test uses sampling just in the nostrils, in the anterior part of the nasal cavity. So that is not unpleasant in any way. I mean, we do it as volunteers within the company for development purposes, regularly. It's not like the nasopharyngeal swabs, which are many of you will have experienced and are very unpleasant and make you gag. So it's very easy to do, and do frequently every day without any discomfort. So it's a test using anterior nasal swabs, 20-minute read time, and is intended to be used to identify individuals with higher viral loads that increase the likelihood of transmitting the infection to others. And the initial CE mark will be for professional use. Within the test kit, as you can see, there is a swab, a packaged swab. There is a foil pouched lateral flow test. I'll show you that in a moment. There is a blister pack in the middle of that image there that contains the buffer, which is introduced into the dropper bottle, the plastic dropper bottle. The anterior nasal area is swabbed. The swab is put into the dropper bottle, into the buffer, and then the cap is put on. You follow the instructions to swish the buffer around and remove the virus from the swab as efficiently as possible, and then several drops according to the instructions are added to the lateral flow test. So that's the actual product. Now, when we think about the performance of a diagnostic test, clearly clinical performance is ultimately the the indication of the how good the product is. But we start off doing what's called analytical performance. So this is something we can do in the laboratory before we get to patients, with what we call contrived samples. So samples that are representative as closely as possible of the clinical samples ultimately. And what we've done here is use on the left, to look at the sensitivity of this test, is to use volunteers in AVACTA negative noses, if you like, and to swab those noses and then actually introduce the viral antigen, the spike protein, into those samples. So they're not real positive samples, but they are quite representative of what the real sample will look like. And what you see in the graph are 2 sensitivity curves, one for a 10-minute read and one for a 20-minute read. So you can see actually that The test works perfectly well with a 10-minute read, obviously until you get down to the very lowest levels of concentration of the virus. It's a little bit small to read, it's on a log scale, but just to let you know, that lowest data point is at 50 picogram per mil of spike antigen. And to put that into context, without going into lots of detail, that is a very low concentration of spike antigen to be able to detect. The 20-minute read, clearly there is an improved signal, and if you look at the blue line, that's the limit of visual read. I'll show you some pictures in a moment, but that's the limit of the visual read, because obviously the less antigen there is, the more faint the test line becomes. With a 20-minute read, it's quite visible. With a 10-minute read, it's right on the borderline at that lowest level. So that little bit of extra information there for you on the analytical sensitivity. The specificity, in other words, does it only detect the SARS-CoV-2 spike antigen? Does it not cross-react with other viruses? There's half a dozen there common closely related coronaviruses. We've also shown that the test will detect the B117 or Kent variant, as it's known, which is prevalent in the UK. The D614G variant, which is prevalent in most of the world, actually. Actually, hot off the press, I can tell you that the test also detects the South African variant. So that's data just in. So some examples of the test having been run. So there's 10 examples here. These are from clinical studies. On the left, positive results. So hopefully on your screens, you can clearly see the test line, which is the bottom line, the first line, labeled with a T on the plastic there. Clear positive signals. The top line is the C line, the control line, and that indicates that the test has run correctly. So for a positive result, you need to see both of those lines. And on the right-hand side, 5 negative results with no test line, no T line visible, but the control line should appear and does, telling you that the buffer and the sample have actually run through the test, and it's been run properly. Okay, so then on to the clinical data, and I think last time we presented, we were talking about the clinical data as being the major hurdle, the major remaining risk, of course, to the development and commercialization of the product. You will have seen on Tuesday we announced certainly the top line data, a bit more information here that I can share with you, but the data are excellent, I'm pleased to say. And we did also benchmark our lateral flow test against 2 other marketed lateral flow tests. So the positive samples you see here, 98 positive samples again across a broad CT range. We benchmarked all 98 of those samples against a leading lateral flow test, which I can't name. And we also tested 10 samples, a subset of 10 samples against all 3, ours, the one I've just mentioned, and another one that's very familiar to us all. So on the left-hand side here, you see the PCR result. So the top box being less than 27. So cast your mind back to 27 being a reasonable level of, to identify infectious individuals, and then going down that column, higher and higher CT values, in other words, getting lower and lower viral loads. So below 27, we had 52 patients in this study that had CT values of less than 27. We got all 52 of those correct, which within that, in air quotes, infectious viral load is 100% sensitivity. And then as you increase the viral load, you can see one was actually missed there between 27 and 28, and one was missed in the low 30s, giving you a cumulative overall sensitivity across that broad range of 98%. The bottom table shows you the negative results, 102 negative samples confirmed by PCR. 101 out of those 102 confirmed as negative, giving you a specificity of 99%. Obviously, if you only do 100 samples and every sample is 1%, so if we did 1,000 over time, we will do more, and I'm sure we can be more accurate with that decimal point as well over time. So on to manufacturing. So we have had, as I'm sure many of you will know, a close collaboration since autumn of last year with Mologic. And to give that collaboration credit, it was instrumental in arriving at the final architecture that we've just been talking about. So we've worked with a number of partners, both disclosed and undisclosed. Actually, we worked broadly with quite a few partners to get the best possible test that we could, and all of those projects contributed to the final test architecture. But Mologic, that collaboration was instrumental. They are closely linked to a company called Global Access Diagnostics, which is a manufacturing LFT manufacturing business. So they were already in an advanced state of readiness to manufacture this particular test architecture. So GAD have already manufactured first commercial batches and have been making product for our clinical validation and verification studies, which I'll explain on the next slide. Abingdon and BBI are now adopting those manufacturing processes from GAD, and there are other UK manufacturers that we certainly would hope to access, as well as potential overseas capacity. So within the UK, we expect to be able to access fairly shortly during the summer around 5 million tests per calendar month. I can't be accurate at the moment as to what the upper limit of that is, because there is a lot of work going on through government investment and other sources to increase the UK capacity. But to give you some guidance, I think that number could easily reach 30 million tests from UK manufacturers this year. But it does depend on a lot of things. So I'm sorry, that's a broad range, but that's the that's the best I can do without being misleading at the moment. So we are, as we'll talk about in a moment, we are very pleased with the demand that we are hearing about, and I'll talk about that in a moment. So we are more concerned about product supply now, certainly, than demand. So we are looking at overseas manufacturing capabilities, as well as focusing primarily on the UK to be able to meet that demand as we ink deals. So to talk through that timeline to launch, which now I can give you a really accurate picture of that. We've passed through the clinical validation, which was clearly an unknown. There's quite a large number of patient samples there, so that's a robust trial going down to low viral loads, high CT values. So it's a good set of data for the CE marking. We have to complete what's called the verification, which, without going into lots of detail, is more analytical measurements for the final technical file. But the rate determining factor is stability. So obviously shelf life of the product effectively. You don't do that in real time only, because otherwise you would have to wait many months or a year to get a year's shelf life. So you do that as an accelerated study. So you raise the temperature, and then, without boring you with the details, there's a way to extrapolate from the accelerated data to the real-time shelf life. That will be completed by the end of April, allowing us to CE mark for professional use very early in May, and immediately begin the commercial rollout in markets that accept the CE mark for professional use. So from a commercial perspective, we've made over the past 6 months or so, we've made really good progress with distributors, end users, OEM partners, and so forth. Obviously, they have been waiting to see the full clinical validation, which, as we've seen, is excellent. So that has allowed us to move on with those discussions that we've got in place quite quickly now. We've got an ongoing dialogue with the Department of Health in the UK. So the CE mark obviously covers Europe and the UK. At the moment, the Department of Health is providing tests free of charge in the UK. So from a commercial perspective, that doesn't leave any room for private industry to sell tests, really. So the Department of Health is the commercial route to market in the UK. So that's important. We've got a very open and ongoing dialogue with the Department of Health, and I will update you on that when I can. We are working in parallel towards CE marking the test for consumer self-testing. So that requires an additional lay user study and a more stringent external audit of the technical files and lay user study. There isn't time to go into that here, but we're also looking at the US regulatory strategy as well. Just to remind you, we are aware of significant demand in Europe. Thinking about the US market, we also have to think about supply. So that would require us to get additional manufacturing capacity in place. But that's all happening in parallel as we speak. And then I just wanted to catch everybody up on a couple of other things, COVID and non-COVID related, actually. The work we've been doing with Adeptrix and Bruker. So with Adeptrix and Bruker, we've developed what's called a bead-assisted mass spectrometry assay for the COVID infection. We have been in clinical studies for several months now. At the moment, the access to positive samples is quite low. It's very low, but we have quite a bit of data now around that test. The thing that has changed, quite significantly, and we've seen PCR contracts not being extended and so forth, because the PCR capacity now is very well established and satisfied. So there is our original intention was to provide mass spectrometry testing as an additional capacity, because the hospital mass spectrometers are not being used for COVID testing, weren't being used for COVID testing. That's changed quite significantly now. That additional capacity is not required. So we're looking closely with Bruker and Adeptrix at the commercial strategy for this test, and actually a wider group of tests on the BAMS platform. So I'll update you on that and the wider, the wider commercial opportunity with Bruker and Adeptrix when I can. In other news, we developed an Elisa test, which, for those who don't know, is a laboratory research test for studying the SARS-CoV-2 virus via the spike antigen. And we secured a distribution and marketing deal with Abcam a few weeks ago. So that's obviously, I mean, they are the big beast in the room in terms of supplying research ELISAs. So that's a great partner to have on board. It's non-exclusive, actually, so there will be other partners on board, but obviously Abcam is one of, if not the best partner to have for that product. And then finally, a non-COVID piece of news. You're all aware that we're focused intently on the diagnostic applications of the AFAMA platform in our diagnostics division now, but there are numerous other applications that we have worked on over the past 3 or 4 years, and one of them is in purification systems, so what's called affinity chromatography. Astrea Bioseparations is a company that works in that space and has evaluated the technology over a long period, actually, in one particular application. And they approached us wanting a license to the AFAMA platform for affinity separation for bioprocessing. So we were very happy to do that, actually, because it's not an area that we're going to exploit in-house. So it gives us an opportunity to monetize that, the AFAMA platform in that application off our balance sheet. We've initially made that a non-exclusive agreement, but if Astrea meets certain commercial performance criteria, then we can take that, or they can take that that agreement exclusive. Okay, so with that, I will hand over to Neil Bell, who will take you through the precision platform, and in particular AVA 6000, which is another major value driver for the group this year. Neil.
Great, thanks, Alistair. And morning, everyone. So this morning, I'm going to actually share a high-level update on the progress we've made to date with our first precision development candidate, AVA 6000, and our plans to take it into the clinic by the middle of this year. And I'll start by giving you some background on the precision platform itself. So what is precision? Well, many of you know that chemotherapy agents exhibit very good efficacy, but actually they also exhibit serious toxicities which are associated with their treatment in patients, and this actually limits their clinical utility. So AVACTA's precision technology has been specifically designed to take these agents and target them more precisely to the tumor, whilst limiting the serious toxicities to healthy organs and tissues. The technology itself comprises of a substrate designed as a dipeptide, which is sensitive to cleavage by an enzyme which actually resides within the tumor microenvironment called fibroblast activation protein alpha, FAP. This enzyme is highly expressed, highly upregulated in the tumor microenvironment of most solid carcinoid tumors compared to normal healthy tissues. The dipeptide substrate itself is actually chemically linked to the chemotherapy agent, and the linked drug is actually prevented or limited from entering cells by this linkage, and it renders the chemotherapy agent inactive. So this is, as we infuse this linked chemotherapy into the body, it distributes widely, it remains inactive until it reaches the tumor site, where it is activated by high levels of FAP enzyme, cleaving the linker from the chemotherapy agent. So just to give you an illustration of how we're actually doing this, because what's really important is that we need to have specificity around this technology for it to work. So the graph at the top right hand of the slide here actually illustrates the localization of high levels of FAP expression in the tumor and across a number of tumor types here in this graph, actually compared to their matching serum levels, which, as you can see, have very low levels of FAP at all. So in addition to that, we need to be confident that the substrate is not removed by other closely related enzymes in the body. And the graph on the bottom left here on the slide actually illustrates the specificity of the substrate to the FAP enzyme, where related enzymes have been shown not to remove the substrate, and this actually is an important component in the technology. And finally, just to say that we have actually licensed this substrate exclusively from Tufts University in the US. We're working very closely with our colleagues in Tufts to accelerate the next generation of FAP-activated chemotherapies. So hopefully you can see the next slide, which is about AVA 6000. So this is our pro-doxorubicin, FAP-activated pro-doxorubicin. So although doxorubicin has been around for many years and is a very effective and widely used chemotherapy agent for the treatment of various solid malignancies, its clinical utility is actually limited by the dose-limiting toxicities, which include cardiotoxicity and myelosuppression, that's suppression of the bone marrow. They're two different mechanisms of action, of course, but doxorubicin's cardiotoxicity effects are well documented in the literature. Doxorubicin causes cumulative and dose-dependent damage to the heart, ranging from hidden changes in the myocardial structure and the function, so acute changes, through to late changes, which actually manifest themselves as cardiomyopathy, which is enlargement of the heart, leading to congestive heart failure. So consequently, doxorubicin's use is limited in patients to a maximum lifetime cumulative dose of 450 milligrams per square meter, which in turn actually limits the maximum number of treatment cycles to 6 treatment cycles every 3 weeks at doses between 60 and 75 milligrams per square meter. So this actually means that many patients don't actually receive further efficacious doses and have to be withdrawn from this treatment. In addition, doxorubicin is contraindicated in patients with significant heart complications. So again, a number of patients don't even receive doxorubicin. So AVA 6000 actually has the potential to address these key cardiac safety concerns for doxorubicin. And 6000 consists of doxorubicin chemically bonded into the substrate and the dipeptide leaving group, which is designed to be cleaved specifically by the FAP enzyme. And as a consequence, 6000 has the potential to deliver doxorubicin directly to the tumor microenvironment while exposing the patient to a much reduced degree of doxorubicin-associated toxicities. And data from mouse models have demonstrated that this differential accumulation within the tumor compared to the heart tissue at high doses of AVA 6000, actually compared to doxorubicin. You can see this in the slide on the right here, where at 12 milligrams per kg of AVA 6000, we can actually see 18 times more active chemotherapy in the tumor compared to the heart. So moving on to our first phase 1 study in AVA 6000, I think it's important to note that at this point that this will be AVACTA's first entry into the clinic and will transform AVACTA into a clinical stage company. In terms of our first clinical study, first into human, we, from a regulatory perspective, we've filed the clinical trial application to the MHRA. We did that back at the end of December 2020, and we have received the CTA approval on the 16th of February, which is actually 48 days after submitting, which was extremely good. We've also received our Research Ethics Committee approval as well, called a favourable opinion in the UK. And we're now actively preparing our sites and our vendors to prepare for the dosing of our first patient, which is currently planned for mid-year. The next important milestone after first patient dose will be the review of the initial safety and the pharmacokinetic data from our first dose and our first cohort. And that will be in Q4 of 2021 at the end of this year. And so the drug level data, which we term pharmacokinetics, will provide further insights into the precision platform and will help us validate the FAP targeted platform. So just to talk about the study itself and the design, the phase 1 clinical study has been designed in 2 parts, A and B. Part A is a dose escalation, where the main objective of the study is to assess the safety and the tolerability of AVA 6000 and to determine the maximum tolerated dose and the recommended phase 2 dose to take into Part B. We're anticipating that Part A will recruit approximately 15 to 20 patients through 4 dose levels, depending on the dose-limiting toxicities seen in the study, of course. And patients will be recruited across a wide range of cancer types. Part B is a dose expansion stage, where the main objective of the study will be to assess safety and tolerability, but also to look at a larger cohort of patients in possibly 1 to 3 tumor types. And bearing in mind that we remain in a COVID-19 pandemic, so we have actually performed a COVID-19 risk assessment. We've shared that with the MHRA. We've shared it with our investigator sites. And we will ensure that we are trying to remain safe, but at the same time actually moving the study forward to recruit patients. I also intend to, or we also intend to include US sites, possibly in the dose expansion study, and we're also preparing an IND for submission to the FDA as well this year. In just focusing down on the study design itself, it's a classical 3+3 design for Part A, where 3 patients will be dosed, and if no dose-limiting toxicities are observed, we will then escalate to the next dose level until we reach maximum tolerated dose. If we do see dose-limiting toxicities, we will expand the cohort by adding further patients. The starting dose for this study is actually set at 18 milligrams per square meter, which is equivalent to 54 milligrams of doxorubicin administered every 3 weeks. And subsequent doses of AVA 6000 will follow what we call a modified Fibonacci series. It's a mathematical series, but it will also be guided by the safety data and the pharmacokinetic data from the cohort of patients. We do aim to dose the patients up to 6 cycles of AVA 6000 if we can, every 3 weeks. However, we've written the protocol in a flexible way, so we do have the opportunity to actually go beyond 6 cycles if we can. So just talking about the wider precision prodrug opportunity, the wider opportunity to apply the precision technology across a range of other chemotherapies is compelling. Many chemotherapies do have dose-limiting toxicities which restrict their utility in patients. And yet, despite these toxicities, these chemotherapies still retain significant market share and continue to increase their share annually due to improved cancer detection rates. So as an example, Takeda's Velcade is a protease inhibitor, and although it's an effective therapeutic agent, its true potential has never really been fulfilled in the market due to dose-limiting toxicities. And these toxicities mainly are damage to the nerves, a condition called peripheral neuropathy. And Velcade only has 2 indications: multiple myeloma and mantle cell lymphoma. Taxanes are another example, such as paclitaxel. Paclitaxel has significant sales potential through to 2025, but its market share is constrained due to toxicities, again, such as myelosuppression and peripheral neuropathy. So the precision platform has the potential to modify these agents and to move the risk-benefit scale towards increased efficacy and reduced toxicities, and allowing these agents to be further exploited in the cancer field within a wider set of patients and cancer indications. So in summary, we're at an exciting crossroads with precision and AVA 6000 development. And with our newly formed development team in Cambridge, we have the opportunity to really drive multiple assets into the clinic towards the key milestones within the development plan. So I'll stop there and I'll hand back to Alistair.
Thank you. Thanks very much, Neil. So just one last slide and then a summary. We can get to the questions. So whilst we've talked there and focused on what I think is the major value driver for the group this this calendar year and dosing first patients in the middle of the year, we have managed, despite the COVID restrictions on laboratory working and social distancing and so on, we've managed to keep our other in-house programs, which we don't have time to talk about, but our partnered programs as well moving forward. So as Tony mentioned, we signed a new deal at the start of 21 with Point Biopharma. Point is a radiopharmaceutical biotech. So the concept here, this is a license for the precision technology that Neil's just described to you. And this is the application, completely analogous application of the precision chemistry, the substrate, to radioligands to improve the targeting to cancer cells. So that first program has been initiated. The LG Chem Partnership focused around autoimmune and oncology. We expanded that partnership last year to include the Affimer XT serum half-life extension, and that program, its primary focus at the moment is around a serum half-life extended PD-L1 Affimer antagonist to be taken forwards by LG. ADC Therapeutics is a let me call it a traditional antibody drug conjugate company, particularly focused on using the PBD warheads that it licensed from AstraZeneca. What our work with ADC Therapeutics focuses on is making Affimer, traditional Affimer drug conjugates that are internalized into cells and the warheads released inside the cell. So we are generating targeting Affimer proteins for ADC, who will do the preclinical research and hopefully take those forward into the clinic themselves. And finally, De Jong, which is a really exciting collaboration around engineered cell and gene therapies with an initial focus on mesenchymal stem cell therapies that have been engineered to express and secrete immunomodulatory Affimers in the tissues where the stem cells end up. So these are, to put it really crudely, turbocharged stem cells that also deliver an Affimer immunotherapy to support the stem cell activity. And we raised funding for Series A funding for AFIXL, the joint venture, primarily from Asian, South Korean VCs, and we anticipate seeing in vivo proof of concept for at least one of these therapies in 2021. So the final slide, in summary then, and I won't read through all of this, we have some time for questions. Clearly there's going to be an ongoing need for a considerable amount of time for high quality rapid antigen tests. We've seen now really good performance of the AFIDX SARS-CoV-2 antigen lateral flow test in that infectious range, which is obviously debatable, as I said many times. This is biology, so there's no black and white infectious range, but overall a broad range of CT values, 98% sensitivity and 99% specificity. So we'll CE mark that very shortly and begin the commercial rollout in May. We have good visibility now on demand in a number of European countries and the UK. And as I said, I think during the presentation, our preoccupation within the company is on supply meeting demand. So overseas capacity, I would love to manufacture everything in the UK and keep all that value within the UK. We may have to look at overseas manufacturing in Europe and elsewhere. From a therapeutics perspective, Neil's talked you through AVA6000. I mean, this is an enormous step forward for the company to become a clinical stage company. We'll see before the end of the year the initial PK data that Neil described from AVA6000, which if that is positive and it reflects the really good data we've seen in preclinical models, then we have not only a valuable asset on our hands in terms of a prodrug form of doxorubicin, but we open up that pipeline that Neil described of future prodrug precision chemotherapies. Strong balance sheet, as Tony described, so we are fully funded through well into 23, and that's not including the contribution that COVID test sales revenue will make to the balance sheet as well. So I will leave it there, and we've got 10 minutes or so for questions.
Fantastic. Alistair, Tony, Neil, thank you very much indeed for the presentation. I would like to remind investors the recording of the presentation, along with a copy of the slides and the published Q&A, can be accessed via your investor dashboard on the Investor Meet Company platform. I'd also like to remind you that your feedback is important to the company, and immediately after the presentation has ended, you'll be redirected for the opportunity to provide your feedback so the company can better understand your views and expectations. Alice, just moving on to the Q&A. Obviously conscious of time, we've received a number of questions from investors, and perhaps I can just start the session by picking up on several of these. I'd also like just to remind investors that due to the number of attendees and questions, we obviously won't have time to go through everything in the session. However, the company will review all questions submitted today. Alice, so the first question we've got here reads as follows: will the huge quantities of lateral flow tests be recycled? If yes, how will they be gathered and processed, and by whom? And that's from Eddie.
Okay, thank you. No, that's a really good question, and actually something that we have discussed internally and been acutely aware of. And actually, not just AVACTA, by the way. Other companies that I've spoken to are acutely aware of the plastic waste that is going to be produced with the sorts of volumes of testing that are being talked about. So I can't say we have made a huge amount of progress in that regard, Eddie, but we have got some initial thinking around biodegradable plastics. Obviously, we would want to introduce that as quickly as possible. So we are aware of that. We are not ignoring the issue that we and many others, of course, are already causing. But I don't have an answer yet about who would collect those and process those. If we can get to a biodegradable plastic, that would be I'd be really pleased with that.
Thank you, Alistair. Next one we've got here is: why did the saliva test fail? What were the technical reasons for the failure to detect or manufacture when there are about 4 other working saliva tests internationally?
Okay, so we took I mean, it's not correct to say it failed. It didn't fail. We took a decision not to progress the saliva test for several reasons. So back in, I think it must have been around September time last year, maybe the end of September, early October, we had got the saliva version of the test at that stage working, as well as the anterior nasal swab working. Excuse me. And the as has been, the shifting sands throughout this entire process, the market changed, and certainly the UK government and other end users moved away from saliva, primarily because of the very complex biology of saliva. Not only the variation throughout the day, but the complexity around what people had eaten, drunk, or otherwise put in their mouths. So it became, for end users, a less attractive sample, although I think we all viewed it as a really attractive, easily accessed sample at the beginning. So because of that, we focused on the anterior nasal swab. So it's not correct to say that it failed. It worked. We didn't take it through detailed analytical testing at that point because we dropped it for commercial reasons. So it's still there if we wanted to use it, perhaps not for COVID, but for other testing, because getting saliva to work was not easy. As you know, there are not many tests out there that outside of COVID based on saliva. So.
Thanks, Alistair. You did pick up on this in the presentation, but we've had a number of questions through. But just to reiterate, does the lateral flow test detect the South African COVID variant? Yeah, good question. Yeah, we did. It was a stop press during the presentation. Yes, we did. It does pick up the South African variant. Great, thank you. I've got here, it was briefly mentioned that the LFT framework is now plug and play with new affirmers for variants viruses. Practically, how will this process work, and how often do you envisage that having to happen? Yeah, okay, so that's referring to if we come across significant mutations, variants that are not detected by the test. So both the spike protein and the nucleocapsid protein are mutated, but the spike protein generally is mutated more. So what you can see is that we are clearly binding to an epitope on the spike protein, sorry, a region of the spike protein that is not mutating, because we can detect all of those variants. So at the moment, the data make us confident that that's not going to be required, but the real benefit of the AFIMA platform, and actually thinking forward about future pandemic preparedness, we generated that suite of AFIMAs to bind to the spike antigen in a matter of weeks. I think it was 4 or 5 weeks. So in terms of responding to changes or future epidemics and pandemics, now that we have a test architecture that works really well with nasal samples and saliva, as I just pointed out,
then it's a relatively quick process to identify a new ephemery agent that identifies the new variant or the new virus, and to then incorporate that into the test by putting it onto the beads that give you the visible readout. So fairly straightforward. And I think going forwards, pandemic preparedness is something that AVACTA should position itself at the centre of.
That's great. We've got a few questions just around BAMS. I'll just put these together. Has BAMS been shelved at this point? And assuming an update on BAMS is not within the presentation, obviously a pre-submitted question, could you please give one to investors and explain where you are with the clinical evaluation in the UK hospital to be in progress in the RNS on February 24th?
Yeah, okay. I think we probably touched on that in the presentation. So certainly not, BAMS has not been shelved. We've got an active collaboration with Brook and Adeptrix, but both on the, I mean, Brooker presented around the COVID testing quite recently. The question now is whether there is a significant commercial pull for us to roll that test out as an IVD or perhaps a research tool or in some other form. But BAMS and the wider context, proteomic testing is is still of great interest to all the parties involved. So I think we touched on that in the presentation.
And again, I think the CE marks, we've had a number of questions around the CE marks, but two questions really sort of pulled together, and you have covered it off. But just to really clarify for investors, when are you expecting the CE to be completed for the test, and how soon will a home use CE mark come after a successful professional use CE mark?
Yeah, so we've got clarity now. I think we can confidently say early May for the CE mark and commercial rollout. We are working in parallel on what's called the consumer self-testing regulatory process in Europe and the UK. I don't have a clear timeline on that. Clearly, it has to be relatively quick in order for it to be useful in the pandemic situation that we're in. But it requires other studies and requires an audit by an external party. So I can't give a precise timeline, but it has to be this year in order for it to be useful.
And just on production, are Abingdon, BBI and Mologic still partners for the production of LFT? Nothing's been said with regards to Abingdon and BBI for some time.
Yes. So the again, touched on that in the presentation. The architecture of the test that we finally ended up with that performed best and clearly really well was through collaboration with a number of parties, including Mologic and GAD, because they are closely working with Mologic on other products. We're ready to manufacture that very quickly, so we have tapped into that, which is great. And now Abingdon and BBI are in the process of adopting those GAD-driven processes. So they're a little bit behind because the manufacturing process changed slightly, but on the way.
That's great, thank you. And one final one we've got here is, which hospitals will the cancer trials be held at?
I'll let Neil answer that one.
Okay, so it's a good question. We've been very careful about selecting the investigators in this study because we wanted the people who are really experienced in first into human studies, but also we wanted to work with people who had experience of prodrugs and doxorubicin in particular. So we're working with, starting with Chris Twelves, who's our chief investigator at Leeds. We're working with the Royal Marsden in London. We're working at the Freeman Hospital in Newcastle, at the Christie in Manchester, at the Beatson in Glasgow and Sheffield. So each of these hospitals and each of these investigators are incredibly excited about this study. We'll also be putting this study onto clinicaltrials.gov onto the US website for clinical trials information, and it will also be on the EU database as well.
That's great. Thank you very much, Neil. And Alice, just coming up to the hour, so perhaps if I could just ask you for a final few words just to wrap up before we redirect investors to give you some feedback, please.
Yes, thank you. So I mean, first of all, thank you for your time, for sparing an hour to listen to the results. Also, I'd just like to acknowledge the effort and commitment, despite quite difficult working conditions, that the staff in AVACTA have displayed over the last year, both in Cambridge and in Wetherby, to deliver what's been I think we can call it a transformational year, without doubt, for the group, and set us up for some very significant commercial revenue-driven milestones around COVID testing, and the huge step into being a clinical stage company this year and through into subsequent years. So thank you to the audience, and thank you to all of our staff who've contributed to that. And we'll look at your, I mean, there are many questions, so we'll look at those and come back to you on those we can in due course. Thank you, everyone.
That's great. Elisa, Tony, Neil, thank you very much indeed for updating investors today. Can I please ask investors not to close the session? You should be automatically redirected for the opportunity to provide your feedback. If you've accessed the meeting from our website, the feedback page will appear in front of you. If you've accessed the meeting via the link sent you in an email, you'll simply be asked to log back in and to submit your feedback, which will be greatly received by the company. On behalf of the management team of AVACTA Group PLC, we'd like to thank you for attending today's presentation. That concludes today's session. Good afternoon.